All right, are you good to get started?
Yep.
Perfect. So hi, everybody. Thank you for joining the Marine Institute Graduate Seminar Series. We meet every other Thursday at 2:30 Newfoundland time. This is being recorded and will be uploaded to YouTube channel where you can find almost all of our other seminars from the last couple of years. So before we start, I'd like to provide the land acknowledgement. We respectfully acknowledge the territory in which we gather as the ancestral homelands of the Beothuk, and the island of Newfoundland as the ancestral homelands of the Mi'kmaq and Beothuk. We would also like to recognize the Inuit of Nunatsiavut and Nunatuckavut, and the Innu of Nizhasian and their ancestors as the original peoples of Labrador. We strive to respectful relationships with all the people of this province as we search for collective healing and true reconciliation and honor this beautiful land together. Now, join me in welcoming today's seminar speaker. Christian Petitpas is here with us from the Massachusetts Division of Marine Fisheries, where she is the Regional Supervisor of Massachusetts Division of Marine Fisheries, Shellfish and Sanitation Management Program in New Bedford. Dr. Petitpas earned her Ph.D. at the University of Massachusetts Dartmouth School for Marine Science and Technology, and her doctoral research focused on marine harmful algae, specifically the dinoflagellate responsible for paralytic shellfish poisoning. Today, Dr. Petitpas oversees private and municipal marine aquaculture in Massachusetts. She works closely with the Massachusetts Department of Public Health in the development and implementation of the Massachusetts Vibrio Paramylacus Control Plan. She is also integral in developing and implementing the state's Biotoxin Management Plan and oversees shellfish growing area classification in Massachusetts South Shore, Cape Cod, and Islands. So please welcome Dr. Petitpas.
Okay, thank you very much, and thank you, Anat, for the invitation to present today. So I'm going to talk a little bit about Vibrio parahemolyticus, which is a naturally occurring bacteria, how we deal with risk in Massachusetts, and also a study on some temperature abuse in cultured oysters. As my introduction just informed you, I'm with the Massachusetts Division of Marine Fisheries. So my talk's going to go over a little bit of Vibrio background. I'm going to talk about the management of Vibrio and the risk in Massachusetts, and then we'll go over the cultured oyster temperature abuse and resubmergence study, and I'll get into what we mean by the temperature abuse and resubmergence and why it's important for us to do that type of study and find out the impacts of resubmergence. So if you've never heard of Vibrio, it's a naturally occurring bacteria. It's a genus that is very diverse, has lots of different species, and they're free living and associated with humans and animal hosts. They tend to like salty water, so they're halophilic, and like most bacteria, they like high temperatures. So when it's summertime and you're getting those high growth rates, in fact, it has one of the highest growth rates of all known bacteria. About a dozen of the many species can cause human illness known as vibriosis, and of the species that can cause the human illnesses, only there's a limited amount of strains that are associated with the illnesses compared to the total Vibrio parahemolyticus in the water. So Vibrio parahemolyticus is actually the species that we're most concerned about in Massachusetts, and we use the shorthand VP. And we're most concerned about VP relative to the consumption of shellfish, and more specifically oysters, because the vast majority of oysters harvested in Massachusetts are harvested for the raw half shell market. So we have people consuming an uncooked product, and as filter feeders, they can concentrate contaminants, including the VP bacteria, up to 100 times greater inside their tissues than what exists in the water column. So every year we get approximately 80,000 illnesses, and about 45,000 of those Vibrio illnesses are VP specific. And in the United States, about 500 hospitalizations and 100 deaths occur per year. So VP is the leading cause of seafood-borne illness in really globally. But 87% require no medical attention. You know, I think most of us have had a stomach bug. We're miserable for a couple of days, but we get over it, and it's the typical gastrointestinal distress. But severe illness can happen in people who are already compromised. So the old, those that have weakened immune system, diabetes, some kind of secondary health condition. And again, as I said, they're associated with the consumption of raw seafoods, specifically or more particularly oysters. And illnesses have been increasing in the last couple decades, and it's been an ongoing public health challenge and also a burden to industry as we regulate them to try to minimize the risks associated with this public health issue. So Vibrio has been a nationally notifiable disease since 2007. COVIS is the national surveillance system. Cholera and other Vibrio illness surveillance is what the acronym stands for. And it's used for health departments to report the infections from the species, the family of Vibrio, Vibrionaceae. And within Massachusetts, we have our own system for reporting and tracking illnesses called MAVEN, Massachusetts Virtual Epidemiologic Network. And basically, municipalities within the state, their local public health departments or board of health will input clinical cases that have been shown to be positive for Vibrio, and then this system communicates with our Department of Public Health, and then if it's confirmed to be associated with shellfish consumption, the Massachusetts DPH, Department of Public Health, will connect with me at the Division of Marine Fisheries, and we have to do a trace back to try to find out where the shellfish was harvested and if there is increased risk of Vibrio illness associated either with the firm or business that it was harvested by or from the water body that it was harvested from. So Massachusetts, like most states in the US that participate in interstate commerce, have to adhere to the National Shellfish Sanitation Program. It's a cooperative program between member states, the FDA, and industry. And basically it sets a minimum set of standards for the sanitary quality of shellfish and classification of the water bodies that shellfish are harvested from. Okay, so this plot shows pretty much our Vibrio history in Massachusetts. We had our first illness in 2011, and what we have here in the blue line is the number of oyster pieces landed and sold by the million, and then the green line is single source Vibrio cases per million oysters landed. What single source means is that when someone got sick and we did the epidemiological trace back, we could trace it back to a single water body, or what we call a shellfish growing area. And sometimes we don't, because sometimes when you go to a restaurant, you get a medley. You have different brands of oysters. We might get and there may be an oyster from Canada on there and an oyster from Cape Cod. And so we don't know which oyster made that person sick, and they sampled all of the oysters. So that's sort of a case where we can't point to a single source. We call it a multi-source. So this figure just shows where we were able to trace it back to a single source. And the reason that's important is because when we have a certain number of single source illnesses tracked to the same water body, it can be elevated to what we call an outbreak, and then we would have to close down that water body, and it would impact recreational harvest, the commercial harvest, and any aquaculture that may be going on in that water body. So an outbreak was basically 2 or more illnesses epidemiologically linked to oysters harvested at the same time from the same area. And if any state that is a member of the NSSP and putting shellfish into interstate commerce has to adhere to the NSSP standards. And one of the standards is that if you have an outbreak, then you have to have a Vibrio control plan. And so in 2011, we had our first outbreak. And then in 2012, we ended up developing our first Vibrio control plan, but it was only for the eastern side of Cape Cod Bay because that's where our outbreak was happening. And then by 2013, you can see we had a spike in single source Vibrio illnesses per oyster serving. And that precipitated us having to have a Vibrio control plan for the entire state. We have not had any illness outbreaks linked to hard clams. We're hoping to keep it that way because we don't want to have to implement a Vibrio control plan for hard clams. It could be that hard clams are not eaten raw at the same frequency as oysters, because we do have Vibrio illnesses associated with raw clams, but we haven't reached the outbreak level yet. So the primary tool Massachusetts uses for Vibrio control is proper icing. We want to make sure that when the oysters are harvested, they're put into a situation where immediate cooling commences. And so we allow for a couple different strategies for icing. The most effective strategy is a technique of a slurry. And in this picture here, we're seeing a slurry. So they can use potable water or they can use seawater in the growing area where they're harvesting, presuming that their growing area is in the open status, and then ice made from a potable water source. And we can see, especially with the slurry, it doesn't take much for, so the doubling time of oysters is very, very quick when the temperature gets high. So we can see at 70 degrees, their doubling time in the tissue of the oysters is only 3 hours. Once we get over 90, it's an hour. They've doubled their bacteria load in their tissues and increased the risk of getting someone sick. So we do have varying icing responsibilities in different parts of the state. Where we have very high risk, they have only an hour to get their oysters in ice and cooled to a certain temperature. The rest of the state has the 2 hours to icing. Another thing we require as a secondary measure is we always make sure that oysters upon coming out of the water are shaded. So a lot of times people who are harvesting can't take out this big box full of ice, and they may be bringing the oysters in uniced, but we have to make sure they're shaded and make sure ice is allotted. They've applied ice within the time depending on which plan they're on. So from here, I'm going to get into the study we did in 2020, and I'm going to give you a little background on what we saw in the 2020 season when we did this study. So I probably should have given a better locus map that showed where Massachusetts is relative to the United States, but this is Massachusetts, South Coast. The reason I'm zooming in here and not showing you other information is because our north shore of Massachusetts basically has no oyster harvest. So north of basically the Duxbury, Plymouth, and Kingston Bay over here, we have no illnesses because we do not have any harvest. And in the 2020 season, we saw several sole source Vibrio illnesses, and they did come from our hotspots. So 2 of the areas that require 1 hour to icing, not 2 hours, is this Duxbury, Plymouth, Kingston area, and this is our highest value oyster aquaculture area. The other is on the island of Martha's Vineyard in Edgartown. So in Katama, that's another area. We had 8 sole source illnesses come out of Katama in the 2020 season. Total of 19 illnesses. and it varied between sole source, but the majority of them were multi-source. And you can see here, these designations are the water body or the shellfish growing area. And then the only thing I want to point out here is that August 22nd, which is just before we started the experiment or study that I'm going to talk about in a minute, we actually did have an illness from the Duxbury, from the Plymouth area where we did the study. And you can see if you are familiar with the towns, where they were where it was harvested and where it was consumed is, you know, it was harvested on one part of the state and consumed in another part of the state. So these shellfish, even though they're consumed raw, are being transported far distances. I mean, even across the country. And so it's really important to keep them iced to minimize the growth of Vibrio. And actually, I should have pointed out when I was talking about the ice that Vibrio, the icing doesn't kill Vibrio in the oysters. It just prevents further growth after being removed from the water. And the act of pumping the water, as long as the burdens and the concentrations in the water aren't super high, generally the pumping in the water will get them back down to lower Vibrio levels. So now I'm going to talk about I did actually include this. Okay, so, you know, we see Maine, so the United States coast. The experiment was done in Duxbury Harbor, and Plymouth Duxbury Harbor. This particular one was in Plymouth Harbor, which is shared with Duxbury Harbor. And what we did was we chose a site that had both intertidal and subtidal areas. And these sort of aqua polygons, they're all licensed aquaculture sites, and the vast majority of them are growing just oysters, culturing oysters. And so we ended up having a site, a aquaculturist donated product and allotted us space on his site so that we can temperature abuse oysters, making it such that we're increasing the Vibrio load and making them a higher risk of illness. So to assess the impact of 48 hours of exposure, which is typical of air drying practices for anti-fouling purposes. So we'll have aquaculturists that will pull their product out of the water, let it bake in the sun so that the fouling organisms can fall off, and then they'll put it in. But we wanted to see what this practice did on the abundances of total and potentially pathogenic genes in Vibrio. So we did it with oysters from both subtidal and intertidal treatments. We exposed them for 2 days, and then we resubmerged them in their respective locations. And there are Vibrio genes known as TLH, thermolabile hemolysin, which is associated with all Vibrio. It's sort of a marker just for the Vibrio species. And then there are 2 genes that are associated with virulence and causing illness, and that's called thermostabile direct hemolysin, and then we have TDH related. And it's not all that important for you to know that what it stands for, but basically TDH and TRH are the virulence factors, and TLH is basically an indicator that we're dealing with the VP species. So we ended up measuring the initial and then the abused, which would be called day 0, and then 2, 4, 6, and 8 days after we resubmerged them in their respective intertidal or subtidal sites. Now, the intertidal site meant that 2 times a day, those oysters are exposed to the air. So they're not pumping, they're not purging, and they're baking in the sun, potentially increasing their load again. So we would expect to see this cyclic nature of increasing and decreasing Vibrio burdens. And one of the reasons we did this study was because we wanted to know how quickly the shellfish, the oysters, can depurate after being exposed to 2 whole days in the hot sun in the summer. And one of the reasons that's important is because when they do this, we require the growers to resubmerge for a minimum of 10 days. But that was based on a study done on subtidal oysters. And we were wondering, well, the subtidal oysters are always in the water. They're never being exposed within that 10 days, whereas the intertidal oysters are constantly being exposed to the ambient air and not pumping for 2 cycles within the day. So is that 10-day resubmergence adequate for both types of environments? And yeah, we found out that our concerns for the 2 sites were not as expected. So I'm going to spend some time just digesting, like decomposing this for you. I know I violated half the best practices for presentation on a slide because it's so very busy, but I think being able to see everything at once is helpful. So in the top panel, we basically have the environmental conditions. The dark red line shows the air temperature during the period of the study. The dark blue line shows the water temperature during the period of the study. And then the lighter blue line shows the tidal cycle. The pink box within this top panel shows the period of temperature abuse, so it was 2 days. And then the top panel is being referenced by the bottom panel. So where these triangles point shows specifically where the shellfish were harvested and sampled. So at this point on the 24th, and we always sampled at low tide, so you can see they're always pointing to a low tide. We removed the shellfish from the water, and we took some initial background data for when we sampled them first. And so we can see here in the first one, and it's all color coded. So this sort of tan is the pre-abuse, the first sampling, and then it's color coded with day 0, 2 days resubmerged, 4 days resubmerged, 6 days resubmerged, and then the white is on 8 days of resubmergence. And basically what we found, and I should also point out, we ended up having, even though it's not captured here because we subsampled it, I need to update this so that it captures that this day did actually reach 90 degrees. So we got a worst case scenario here where when the oysters were being abused out in the air, we had a 90-degree day, and it's pretty close to worst case scenario in Massachusetts. But basically, we would expect the controls, the pre-abuse genes to be representative of the background. And so we wanted to find out, well, what happens after the abuse and how long does it take to get back to background? And one thing you will see here is, so the TLH, that's the blue bar. The blue bar represents just indicators of total Vibrio. TDH and TRH are indicators of virulence and risk of illness. And one of the first things that kind of stand out at you is we really didn't see too much of the TDH, the high risk virulence factor. It showed up in day 0. So after being abused, we did find it in the intertidal. And so actually I didn't decompose this enough. So within each panel, we have the subtidal control, and then we have the intertidal control on the far left. And then after that day, we have the abused. So on this panel, again, it's day 0. And then we have the subsequent days of resubmergence. And so I know I'm spending a lot of time decomposing this, but the big picture is that we ended up finding a 3-log increase in the virulence factor genes and in the TLH. So total Vibrio increased by 3 log after the abuse. We ended up seeing that the highly virulent gene was expressed in only the intertidal abused, and that we ended up having very, very high loads that would be indicative of illness. We saw that the TDH virulence factor was after the 48-hour temperature abuse was pretty much gone, and then we didn't see it again until day 6. So we're not sure how to explain that. It could just be associated with this, the fact that it's an intertidal area and we're seeing some cyclic nature of it being exposed and then resubmerged. The TRH increased 2 to 3 log and then returned to environmental concentrations on day 6 in the subtidal treatment and day 4 in the intertidal treatment. And so that was sort of counterintuitive to us. We actually expected the subtidal treatment to return to background levels faster because it's not constantly being re-exposed. That was a surprise. The intertidal oysters appeared to purge the Vibrio faster than the subtidal. And there have been several studies that have shown this. And one of the theories is that oysters in the intertidal area have become acclimated to that situation. They know they're going to be exposed, and once they're resubmerged again, they're ready to hit the ground running and start pumping. Whereas for subtidal oysters, it's such a perturbation and such a stun to their system that when they're put back in the water, it may take a little longer for them to start pumping again. So that can be, verified in subsequent studies, but this isn't necessarily a new finding here. But the big picture answer here is that our 10-day resubmergence requirement is sufficient for both types of culture area, both subtidal and intertidal. So just to summarize the results, we had the VP genes. Again, the TLH is total VP, TDH and TRH are virulence factors, and we measured them in initial, abused, and then 2, 4, 6, and 8-day resubmerged samples. The total VP increased a factor of 3 logs in both the subtidal and intertidal, and then they returned to background concentrations by day 8 in the subtidal and 6 in the intertidal. And so that's total VP, not necessarily an indicator of pathogenicity, but still it's something to monitor. But the TDH, which is the more virulent virulence factor gene, we only detected twice. We detected it immediately after the temperature abuse, and then it disappeared, and then we saw it again in the 6-day resubmerged sample. The TRH, which is another virulence factor, just not quite as virulent as the TDH, increased 2 to 3 log and returned to environmental concentrations on day 6 for the subtidal and day 4 for the intertidal. So again, we're finding intertidal oysters are actually doing a better job of getting back to background, even though they're having, with our semi-diurnal tides, constant temperature abuse and exposure to air. And right now, we think our plan is to stay at the 10 days, even though this particular study suggests maybe we could even shave off, a day or 2 from this requirement. I think we need to replicate this in other locations to make sure that this relationship holds in various locations. But we can be confident that the current regulations are sufficiently protective of public health when it comes to these culture practices, such as the air drying for anti-fouling. So with that, I just want to acknowledge that we have partners in that UNH, Steve Jones and Cheryl Whistler. The next steps, which I believe they've already done, but we have to connect with the data, is to actually look at the strains. So what we did was we transferred all of the samples that had that tested positive for the virulence factors, and now they're going to see if it's associated with a specific strain of Vibrio parahemolyticus to find out what we're dealing with. We have found that we have a strain that originated from the West Coast that is a particularly pathogenic strain that we're finding in certain areas on the East Coast. Also, would like to acknowledge NOAA Sea Grant funding, and the ISSC also provided some funding. And also big thanks to our grower, Sean Withington, because he donated the product. And in order to replicate this sample the way this study, each sample was done in triplicate, each sampling session. So it was I think it was 1,000 oysters he donated. And they can get about $1 an oyster. So it was about $1,000 he donated to this project in addition to the space on his aquaculture grant to conduct this study. And so with that, I think I stayed within the half an hour. I'll take any questions.
I have a question. Sorry. uh, with climate change, are you expecting to see any parts of the state of Massachusetts to be not viable for growing anymore, or
Absolutely. You may have heard about this mass die-off on the West Coast a couple years ago, because in the shallow water, in the intertidal, the shellfish were basically baking. So if we have long periods where we're in 90 degrees or 100 degrees, and we have a lot of growers in the intertidal. In fact, on the background on this question page, this is an aerial photograph of, I believe this is Barnstable Harbor, but these are all rows of cages of oysters that are exposed to the air. And if we start getting 90, 100 degree long, heat waves that tend to be, protracted in length, we can see our aquaculture industry having major mortality and losses there. So it could be that people who now have leases in the intertidal or in very shallow water need to rethink and get into deeper water. And of course, with climate change and the changing environment, we could see, shifting distributions of things like Vibrio. I didn't mention there's another species of Vibrio called Vibrio vulnificus that can cause illness. It tends to be more associated with wound illnesses, but that is another bacteria that requires a control plan if we get any outbreaks. And right now, we don't have to have a Vibrio control plan. I'm not positive. I think maybe it's New York or New Jersey is the furthest north that has one. So, we don't know if it's the environment that's select. We know we have Vibrio vulnificus, but why have we not seen any outbreaks? And we don't have a Vibrio control plan, and we don't want one. But it's not clear whether climate change is going to make the north a little more susceptible to Vibrio vulnificus outbreaks. In addition, going and getting past Vibrio, just the other organisms, right? You may have heard of the Florida red tide that caused all kinds of problems. It's killing manatees. We have the Gulf Stream coming up the coast, bringing that water in, and if the environmental conditions, up north end up being suitable for those cells that make their way up there, we could be finding in the next decade or so that we're having, another red tide or harmful algae organism that's sort of wreaking havoc in the north.
I had a question here in the hall kind of related to that. You did mention the first case you saw was in 2011. Was it introduced via climate change, or was it introduced in some other method?
It's not clear. There's a suggestion that it was a specific strain that may have been introduced. The ones that are associated with these pathogenic genes, and it's a combination, I think, of an increase in our industry. If we were to go back to that slide, the number of the number of pieces have basically been growing at a very rapid rate. You can guess what this is, right? This is the pandemic. The pandemic basically closed all the restaurants and decimated the oyster industry. But other than this little blip, we've been growing, and we are now in 2021, we had basically caught up to where we were pre-pandemic, and we've surpassed it in 2022, even though this figure isn't updated. So it's a combination of our consumption habits have been increasing, but I think it also is temperature related. I think there are a lot of factors that are acting synergistically here.
Yeah, that's really interesting. There's so many different parts of all like fisheries and seafood that impact it. Was there any questions in the hall? Yeah, we have a question here. Do you want to come up or do you want me to repeat it? I used to work in the state of Mississippi, and so Vibrio is also a concern there, and we often shut down the fishery, I believe, in the summer. Do you guys have any cases where you have to shut down, or is the control plan enough to keep the fishery open?
So so far it's been enough to keep it open because we haven't had in the last several years any outbreaks, but we have had outbreaks where we have to close them. And so we will, if we can go, and we're surgical about it. So if we have single source illnesses and we can trace it back to a specific portion of the state, a specific growing area, we will close down that area. Also, Massachusetts is unique in the United States. I think there may be one or two other states, but it's a state that has home rule, meaning that the individual municipalities have jurisdiction over their shellfish.
Okay.
And some municipalities choose to not allow harvest because they don't want to deal with having to enforce the Vibrio control plan. Some of them allow the commercial harvest, but not recreational harvest because they don't want the recreational harvesters to just not be, not handle the product correctly, and then they can get sick and then end up shutting down their commercial harvesters. So whereas the commercial harvesters are more regulated, they have to go to certain landing points where they're going to have their icing checked, and they're going to make sure that they have enough ice and they're handling it appropriately. Recreational harvesters are pretty much going where they want, whenever they want, and it's more difficult to make sure that they're taking these protective methods for themselves, precautions for themselves. So it really is a town by town thing where sometimes they just say, I don't want to deal with recreational or commercial. During the Vibrio control season, which I should have mentioned runs from May through October. October 19th is the last day of the Vibrio control season. So from May through October, they have to implement these Vibrio control measures. They have to have the ice, which is a burden. It's a financial burden. It's an operational burden to be lugging the ice out to their sites. And I think having the towns that just don't want to deal with it, you can see where they're coming from.
Yeah, that's interesting. I wouldn't have thought about the recreational fishers as well, for the recreational.
Yeah, and so another thing, another component of your question is when we do the illness traceback, so DMF is more involved in once DPH goes to the restaurants, and because everything has to be tagged so that there's traceability. And so the restaurant should still have a tag, and sometimes if it comes from New York, say, there's an illness, the New York Health Department, they had a clinical test that showed it was positive for Vibrio parahemolyticus, and then their public health will notify our public health, and our public health agents will say, okay, well, what are the tags? And then they'll go to the firm that sold New York that product, and then we'll go out to the growing area. And if at any point in the supply chain we find out that somebody wasn't doing what they're supposed to, for example, we had an illness associated with a Massachusetts harvested oyster in California. And when the agents went to go inspect it, they found out that they were holding the oysters in a refrigerator that was right near the oven. And so every time they opened it, they were getting, hot air that was going into the refrigerator. So essentially, they weren't holding it at the proper temperatures, increased risk. So in that case, we can point to post-harvest abuse that could explain the illness. If there is no post-harvest abuse, we need to assume that the risk is associated with it coming right out of the water. And so in that case, it's associated with the water body, that location, and we'll close it down if there's been an outbreak.
Great. Thank you so much.
You're welcome.
Oh, I think we have one more question maybe in the chat. Yeah. Can you use Vibrio as a biomonitoring tool for other contaminants like PAHs and PCBs?
No, we cannot. I mean, there are other microbes that we use as a monitoring tool. So part of the NSSP is to classify the water bodies or shellfish growing areas. And there are certain classifications that are considered too contaminated to even allow harvest. And so there are certain microbes that we use as indicators. So we use fecal coliform as an indicator, and also something called male-specific colophage, which is a bacteria virus. And so those two things can indicate human sewage, human waste contamination. But as far as I know, I mean, I have not heard of those Vibrio being associated with PCBs. It tends to be more correlated with temperature, and it's not a perfect correlation either, but again, the temperatures go up, their growth rate increases, and illness risk increases.
Amazing. Thank you so much. Anything else? Perfect. Well, thank you so much for your time, Dr. Petitpas, and everyone who joined us today. If you missed anything, this will be recorded and put on our YouTube channel. And our next seminar will be February 9th at 2:30, where we're going to have 4 graduate students showcase their work. So thank you so much, everybody. Have a great day.
Thank you.
Thank you, Chrissy.
Bye, Nat.
Thank you so much.
Bye.